Hi, welcome back to educator.com. This is the lesson on embryological and fetal development. So an overview of development from conception up to adulthood would go like this. It starts with fertilization. That is the moment of conception. When sperm fuses with egg, you're going to get what's called a zygote. That's the first cell of life. Then from that point on, it's embryological development until it becomes a fetus. The first step in getting from zygote to embryo is what's called cleavage, and that's a term for when cells physically divide the cytoplasms and separate. So cleavage happens after telophase, if you took biology recently. But cleavage happens over and over and over and over. You form a ball of cells, and that ball of cells that has a hollow center is called a blastula. And then it goes through what's called gastrulation. Gastrulation is a folding in, and then you get defined layers within that ball of cells. You get outer layers of cells, middle layers of cells, and inner layers of cells. And then there's this gastrula development until we call it an embryo. And it's called an embryo until about week 9, and then from that point on, it's a fetus. And it's a fetus in through the second trimester, third trimester, and then eventually birth happens, and then you have postnatal development. So this baby here, this cute baby, is going to develop, into a toddler, child, adolescence, adulthood, and eventually you reach maturity and you die. Fertilization overview. So how does fertilization actually occur? What are the details? It's fusion of a secondary oocyte and a spermatozoan, or spermatozoa. Secondary oocyte, if you saw the female reproductive system lesson, that comes from what's called a oogonium. That's kind of like the stem cell that makes eggs. And the secondary oocyte is a more technical term rather than just calling it an egg or an ovum. This is actually not completely done with meiosis, and I'm going to cover that a little bit in the future in this lesson. But once the spermatozoan makes contact with it, it will finish meiosis and then become that full-fledged ovum. And each of these should have 23 chromosomes. That's the haploid number. That's chromosomes without a pair that go with those chromosomes. So when you have sperm and egg coming together, you have 23 going with 23 chromosomes, and that's how you get the diploid number, or 46 once again. The volume ratio, when you compare the oocyte and the spermatocyte or spermatozoan, is about 2,000 to 1 volume-wise. So the amazing thing is that when you take one sperm, and if you were to stretch it out with its little sperm head and flagellum, it's close to about half the length of the diameter of an egg cell, but the volume is crazy different. 2,000 times the volume in an egg. And that makes sense because besides, the sperm head having the nucleus, there's not much else going on there. When you look inside of an ovum, one of those egg cells, you've got a lot of cytoplasm outside of the nucleus, but contained within the plasma membrane. All the major organelles that you're going to have in that ball of cells, in that embryo, come from mom, including mitochondria, ER, Golgi, etc. And those are a lot of what are called epigenetic factors, just things other than DNA that are inherited from the maternal side. And fertilization typically occurs within a day after ovulation in the fallopian tube. So if it happens within a day after ovulation, the part of the fallopian tube that is going to happen here is that infundibulum. If you remember, the fallopian tubes have those fimbriae. That's closer to the ovary rather than closer to the uterus. If fertilization happens where the sperm reach the egg in the second day after ovulation, or even the third day, yes, then the egg will have gotten closer to the body of the uterus. And yes, a zygote is formed. Zygote is the term for the first cell of a new life, and that zygote should have 46 chromosomes if all is well. And here's an older micrograph of the surface of an egg cell and a sperm. Oocyte activation. So once sperm actually come into contact with the egg, you're going to have some things happening that are very important in this sequence. So it's a series of events that are triggered when the plasma membranes of sperm and oocyte come into contact with each other. First thing that happens is a block to polyspermy. Here's what that means. It's a fancy way of saying the egg hardens its exterior so that no other sperm can penetrate once that first one has gotten through. When you look at a bunch of sperm trying to penetrate an egg, you have this region on the very outside that actually won't be broken down unless a lot of sperm are poking at it. All of their acrosomes, and you're going to see what an acrosome is later on in this lesson, but all of those little enzyme sacs at the edges of the sperm heads wear away that outer area, and then you get to what's called the zona pellucida. And once a single sperm penetrates the zona pellucida, there's an immediate chemical reaction that hardens the outside, because you don't want multiple sperm entering one egg. You want the diploid number. You want 46 chromosomes. You don't want 92. You don't want 4 copies of every chromosome. That's not going to be a viable human being. So block to polyspermy makes it so that only one sperm head has actually entered the egg cell. Next thing, completion of meiosis II. So remember, a secondary oocyte is not quite a full-fledged ovum. It's actually arrested in meiosis II. So meiosis II completes once that sperm has entered, and then also activation of enzymes. And that increases metabolism. So in an egg cell, you have a lot of mRNA or messenger RNA strands that are just kind of hanging out in the cytoplasm, and they're kept inactive by certain proteins. But once the oocyte is activated by that sperm entering, you turn on the mRNA, and then you start synthesizing a lot of important proteins that are part of making an embryo. So those things are on hold until it needs to start. And once you have that diploid number, once those chromosomes have come together in that zygote, time to get metabolism going, because there's going to be a lot of cell divisions. Only the nucleus of the sperm is supposed to move into the center of the oocyte. You've got that middle piece of a sperm you're going to hear more about later in this lesson, and the flagellum. Those things don't stick around, and a lot of times they fall off right after the sperm has come in. And actually the egg has the potential to destroy those things. If, for some reason, paternal, the dad's side, mitochondria stay around in the egg and they're not destroyed, that actually results in a severe disorder that has a lot of impact on your muscle tissue. So healthy individuals do not have paternal mitochondria. All of the mitochondria that men and women have should be from their mother. And that's because those mitochondria were in the cytoplasm of the egg cell, and the ones from the sperm, they just got the sperm to that destination. They don't stick around typically. Oh, and this is actually a micrograph of an oocyte. They've removed some of the outer nourishing cells, the trophoblast cells that you're going to hear more about later, so you can get a better view of it. But that is an oocyte. All right, cleavage. So cleavage means that it is this pinching in. Let's go through kind of what happens on a day-by-day basis. Day 0, meaning that's the day that fertilization happened. You get the first cleavage division. And then by the time a day has gone by, hey, you've got 2 cells. And the amazing thing is, when we go through all of this, all the way up to blastula, when we get a ball of cells, the size does not change. That's the amazing thing early on, is that you have this large ovum, and then all these cell divisions happen maintained in that space. You don't get a drastic increase in volume, not quite yet. So even when we get to the point where there's 80-something cells, it's taking up the same space as we had at day 0 when there was a zygote. So day 1, 2 cells. 2 cells divide to make 4 cells. Eventually, you get to the point, day 3 approximately, where they say it's an early morula. And a morula is just a dense ball of cells. And then day 4, more cell divisions have happened. Then we have an advanced morula, a little bit more cells taking up that space. And then flash forward to almost a week into this, day 6, we finally have what's called a blastocyst, or blastula is another name for it. And that's what you're looking at right here. This little picture is a hollow ball of cells with a nourishing layer called the trophoblast. Number 4 here, what you're looking at is an inner mass of cells. So I'm going to highlight that in yellow. Inner cell mass, that's what's going to become the embryo, these cells right here. And you'll see what's going to happen to those later on in this lesson. Number 3/2, you're looking at the trophoblast layer around the outside of this. And that is a nourishing body for this very, very young life. And eventually this trophoblast won't be needed, eventually, because it will be connected to the mother's blood supply, but not quite yet. This particular blastula or blastocyst has not anchored itself into the endometrial lining of the uterus. And eventually you'll get a placenta coming out of that and an umbilical cord and all that, but that's not quite yet. Number 1 is called the blastocele. Blastocele, let me spell that out for you. So number 1, blastocele. I know it's a funny spelling, but that's how it's pronounced, blastocele. That is the hollow section in here, and we'll see that even when we get to the folding in, you'll still see this hollow section. But yeah, now we have a blastula, and it's just kind of rolling down the fallopian tubes and into the uterus, into the cavity of the body of the uterus. Implantation. So now this blastocyst is going to implant itself in the endometrium of the uterus. And this usually begins at around day 8. It could be day 9, but day 8 usually. An enzyme is secreted by the trophoblast from that outer layer into the endometrial lining, allowing the initial implantation. So it can kind of penetrate those outer layers of the endometrium. Lacunae, little holes or little channels that help to supply nutrients to the developing trophoblast from the blood supply of the endometrium. And there are these finger-like villi. They look like little finger-like projections extending from the trophoblast into the endometrium, and these continue to develop to start to kind of suck nutrients into this area. I should rephrase that. Initially, what happens is just diffusion. It's diffusion of nutrients, so they're not really sucking it, into the trophoblast region. But diffusion is a passive process, and there's a lot of blood flow from the mother into the endometrium. You're going to see in the next few slides, this is very important. The blood cells that develop in this very early new life, those blood cells, if you look at the hemoglobin of the fetal blood cells, actually they have a higher affinity for oxygen than the maternal blood cells, meaning that the baby's early red blood cells are even more of a magnet for oxygen than the mother's red blood cells. And that's going to be a very important piece of the puzzle in terms of how this works. All right, so after this blastocyst has implanted itself, further development happens within that inner mass of cells. By day 12, the inner cells of the blastocyst have developed into 3 distinct germ layers. Germ here, don't think of bacteria. Think of like the word germination, like in plants, in terms of like a seed, developing and coming to be in terms of it making new life. So germ in that sense, that it's like a germination, new life. So there's 3 distinct germ layers, the ectoderm, mesoderm, and endoderm. Ecto, outer, meso, middle, and endo, inner. And I'm giving you some examples here of what these cells become in a fully developed baby or human being that we can recognize. This is not all of the tissues. You can look up everything that the ectoderm becomes and everything that the endoderm becomes, but these are some major parts. And the way that they know this is by studying gastrulation and gastrula and the development into the embryo and fetus. If you tag certain cells with certain chemicals, and those chemicals remain in the tissues that develop later on, you know that those tissues are descendants from the particular cells that you tagged. And they've done this in animals a lot over the years. So ectoderm, that outer layer that you see here in blue, becomes the epidermis, the outer layers of skin, neural tissue, the brain, nerves, everything, skeleton. Mesoderm, I know you don't see that in this particular drawing, but when this folding in happens from the blastula, you do get like another folding in from this green area. It almost looks like little ears. So that folding in this section here and here is what gives you the mesoderm. The mesoderm forms slightly deeper levels of skin like the dermis. The muscles come from the mesoderm, and your heart and blood vessels come from the mesoderm. And finally, the innermost part next to this cavity, this space here that they call the blastopore. And actually, the archenteron is the term for this. The blastopore is just the opening into the archenteron. My apologies. But the endoderm, you get the digestive tract, glands, a lot of different glands, and the urinary system. The way that I remember that the endoderm gives you the digestive tract is if you think of like the inside of the body, the most inner holes, the most inner cavities inside your body, the digestive system comes to mind. You have this pathway going all through your body from mouth to anus, and the most inner passageways that stuff's moving through, it's easy to think, yeah, that's the digestive tract. And it's funny, this blastopore, we know in humans and in a lot of different animals, vertebrates especially, animals with spines, this blastopore is the first hole that we see in this gastrulation, and that becomes the anus. In other animals, like if you look at insects, the blastopore becomes the mouth. So of course, later on in our development, you're going to see the mouth hole develop, but blastopore becomes the exit for the digestive tract, so it's no surprise that this greenish layer of cells gives rise to the digestive tract. So gastrulation, the forming of a gastrula, is that folding in and establishment of germ layers that become the major tissues of the body. When we look at what happens to the trophoblasts, those surrounding nourishing cells, they become different extraembryonic membranes outside of the embryo itself. Yolk sac is one of them, and this is an interesting thing. We're not birds or reptiles that are born inside of this little egg where you would actually need a yolk sac to nourish you. But the yolk sac, evolutionary biologists assume that this yolk sac is from our ancestors. Mammals came from a class of reptiles, and we are mammals. So yolk sac, it's not filled with yolk. It just doesn't actually contain yolk like you would see in a chicken egg. But it is this sac that helps give rise to our blood cells that eventually are going to receive nutrients, oxygen specifically from the mother through that connection, that placenta we're going to get to in a bit. So the yolk sac, once the placenta is established and the umbilical cord comes to be, the yolk sac is kind of off to the side of the umbilical cord itself. The amnion, you may have heard of amniotic fluid. Yes, the amnion ends up being this fluid-filled region that surrounds the embryo, protecting it. You're going to get cells from the embryo sloughed off into it. Eventually the baby swallows amniotic fluid. But yeah, this is a fluid-filled region that surrounds the embryo, and there's the amniotic sac that contains the amniotic fluid. And as the baby develops, it expands. By the time you're into the second trimester, the amniotic sac has expanded a great deal and eventually is pushing up against the inner edge of the uterus, the endometrium. The allantois, when I first saw this as a student, I thought it was allantois, but it's the allantois is this little area that eventually gives rise to the urinary bladder. And parts of this have to do with the establishment of that connection for nourishment and gases to the mother. The chorion, you see this term chorionic villi associated with chorion. This leads to the entrance of blood vessels feeding the gastrula. So before we have a full-fledged placenta, you have what are called chorionic villi. By the time you're into week 3, in the first month of development, before women typically know that they're pregnant, you have chorionic villi that are helping to get blood vessels from the endometrial lining to feed the baby. At a certain point, the trophoblast is not enough. The trophoblast initially nourishes that ball of cells, but once it grows to the point where it's thousands and thousands of cells, you need a lot more entrance of nourishment and gases from the mother. And chorionic villi have a lot to do with that. There's a test you can do later on in pregnancy called chorionic villi sampling. And this is the tissue that they can get a sample of to test for certain kinds of problems that could be happening with the fetus. So the placenta. Initially, blood supply to the embryo enters through chorionic villi. So initially, we wouldn't call that a placenta, but through the chorionic villi, and those arise from, remember the allantois, the allantoic arteries and veins help that blood connection happen. But by week 5, if you were to go in it with a camera and kind of look around, you would see a placenta. And from that point on, the placenta is there. It grows a little bit as time goes on to help the growing baby. But a full-fledged placenta has some major layers I want to introduce you. So the decidua bacillus, this is a basal layer, and decidua means a falling off, like deciduous leaves, deciduous trees. If you're wondering, well, what's the falling off thing? Look here. This is a little image from Gray's Anatomy. So here's the endometrium. If you're wondering what we're looking at, this whole thing here is the placenta, and here's the umbilical cord. It is anchored to the endometrium of the uterus, and usually it's towards the top, kind of near the fundus of the uterus, towards the top of the body, or superior portion of the body of the uterus. If the placenta is down too low next to the cervix, that could lead to some problems, especially during labor. I'll get to that in a bit. But the deciduous part here, why do they call it this? The decidua, you see here, there's little like breaks in these blood vessels, and blood is just kind of entering this space. That's what this is referring to, that this layer here, parts of the of the maternal tissues that have blood flow, there's kind of a wearing away to the point where maternal blood just pours into this cavity. And that's why I've said, hey, there's a cavity in here. All this space here, you can see it here. It's the intervillous space. You can barely see that term, but this space around the chorionic villi, which are coming from the embryo, you have maternal blood just coming into here. And that's brilliant because think of the chorionic villi as kind of like little blood vessel branches, little blood vessel trees in a sense. And as long as you have maternal blood adjacent to it, diffusion will bring those oxygen gases, the nutrients, into the blood flow of the baby. And what also happens is the waste from the baby enters into this cavity and can go back into the mother. So the carbon dioxide being generated in the baby's body and the waste products from metabolism go the reverse way. The umbilical cord, that's right here. And of course it's cut off, you don't see the full thing, but the umbilical cord goes into the belly button region, what does become the belly button. After the umbilical cord, little nub falls off when a baby's been born. But the umbilical cord has arteries and veins, arteries going into the baby, veins going back to the mother. So here is the placental body. When birth happens, the placenta comes out. They call it the afterbirth, because after the baby comes out and, you know, the baby has an umbilical cord attached to it, what's the other end of the umbilical cord? The placenta. So once the baby's born, there's no need for the placenta anymore. That comes out after the baby. So week 4 embryo, by the time gastrulation has happened and these different areas of that folded in ball of cells become parts that you can actually notice. You know, this doesn't quite look human, but you can see some parts that are going to be more obviously human several weeks later. If we looked at embryos of rabbits, cows, dogs, they would actually look very similar to human embryos at this point in time. But let me show you some of these different parts. So the forebrain, which becomes the majority of the higher brain, that's right here. Forebrain is in this region. The eye, I think you can probably notice the eye already at week 4, right there. The heart, should have used red for that, but I'll use blue. So the heart, right in this region. The pharyngeal arches, oh, I already used green. Purple. The pharyngeal arches, what the heck are those? All right, so here they are. The pharynx is the throat. The amazing thing is that even in a human embryo, you would see these little foldings looking like they could become gills. They don't, because we're human, but this is one of those, as far as we know, vestigial structures, kind of leftover remnants from ancestors in the ocean from hundreds of millions of years ago. I mean, even reptiles, you can trace their ancestry back to vertebrates in the water that breathed with gills. So pharyngeal arches, you would see them for a little while and then they don't develop into gills because, of course, we have lungs and we do not breathe underwater. Arm buds and leg buds, I'm going to highlight those in yellow. Arm bud, leg bud. That's the cool thing is you just see little, they look like little nubs popping out where the arms are going to be and where the legs are going to be. And if you flash forward a few weeks later, you would see a longer bud, and it almost looks like a little paddle for the hand with no fingers. And the way that you get the fingers developing is, imagine that I have no spaces here, and this is all filled in with skin and tissue. What happens is, this region is stimulated to die, this region is stimulated to die. The cells are signaled, hey, you're done. And that's called apoptosis. That's basically programmed cell suicide or cell death. If that does not happen completely, you would be born with webbed hands or webbed feet. So there are places in an embryo where cells need to stop developing to create spaces. And then tail. We actually do have a tail for a while as an embryo. Here it is. You can see that. This, of course, is going to become the spinal column. You can even see already little divisions between the sections of what's going to become vertebrae. But the reason why the tail doesn't look like a tail later on is the amount of growth in this part of the embryo compared to other parts gradually lessens so that it doesn't become a long tail. We grow into that tailbone, so it ends up being not visible outside the buttocks region. All right, a week 8 embryo, about 2 months into the process. This is bigger, of course, than it actually is in real life. The size of a week 8 embryo, about that big. This is typically before a woman is showing, before you could obviously know that she is pregnant. But yeah, about that big, just a few centimeters, about an inch long. This baby, the reason why we have this image is because it could have been a spontaneous abortion. The baby was lost, and this tissue was preserved, probably through the process of plastination, which is a way that you can preserve tissues in a deceased body or an organ from a deceased person. And you could see a very early umbilical cord and how it enters into the body. Yeah, you can see it's about 8 or 9 weeks, 38 millimeters or 3.8 centimeters, close to an inch and a half. Week 12 fetus. By the end of the first trimester, the first approximately 3 months, the rudimentary parts of all major organ systems have formed. The heart is beating. The brain has brain function. There is a liver. There are lungs. Are all of these ready for the outside world? No. The earlier the baby is born, of course, prematurely, the less developed the organs are. So I'm going to talk more about premature births in terms of the danger of that. But you could see with an ultrasound here that this is a human baby, and ultrasounds have gotten better and better over time. They're not all black and white and grainy anymore. There's some good three-dimensional images from ultrasounds. So speaking of ultrasounds, how does that work? Ultrasound is named after the high-frequency sound waves that are used and projected into the uterus, and the reverberation gives an image of the fetus. So the reason why, even though they're sound waves, we can't hear them is the frequency of these sound waves is beyond human hearing, tens of thousands of hertz. Sex can be detected between 18 and 22 weeks. It really depends. But potential birth defects and other problems are also observed. So ultrasounds are not just for telling the mom and dad, hey, it's a boy, it's a girl. Seeing the baby, how it's moving, how the body parts look, can tell the ultrasound technician or a doctor if there are some other problems. So technicians look, oops, that's a typo. Technicians look for obvious genitalia markers to determine sex. So at this point in time, when you're around the 20-week mark or so, you can look at the genital region and see whether or not there's the labia majora. You can see whether or not there's an early penis with the testicular region. All right, flash forward a great deal. Week 40 fetus. That's full-term birth happens at week 40. I was actually closer to 41. I came a little bit later than was expected. But you know, plenty of births happen at week 38, week 39. But week 40, this is an older image of kind of how the baby is situated. A normal presentation, meaning the normal orientation of the fetus prior to birth, is that the head is facing down towards the cervix, towards the birth canal. And you can see that the mother's organs are pressed against a great deal. The intestines, the liver, a lot of different areas are being pushed to the side. And this is one of the reasons that, especially later on in pregnancy, a pregnant woman has to go to the bathroom a lot, because the bladder is being pressed upon, the colon, the intestines are being pressed upon. And also, the stretching of all this material can stretch out the sphincters that keep those things where they're supposed to be. So yeah, by week 40, pregnant women feeling a lot of extra mass there and probably going to the bathroom a lot more. But yeah, now we're going to move on to how does labor happen. So prior to week 40, some women experience false labor. So these are little bits of contractions in the uterine muscles that don't lead to real labor and real birth. So, you know, sometimes it'll be like week 35 or week 36, a woman will feel, you know, stuff going on down there, go to the hospital, and it's a false alarm. The baby's not ready to come out yet. True labor is when biochemical and mechanical factors reach the point of no return. The unconscious portions of the female brain have signaled, hey, it's time, this baby is ready. And labor contractions are what tell you it's true labor. And we're talking, initially, there are 3 stages. Initially, it's dilation. And what's dilating is the cervix. Remember, the cervix is that bottom part, most inferior part of the uterus. And when it starts dilating, it starts gradually opening so that the baby can start to come through the birth canal. And initially, you're going to get you know, contractions that last about a half a minute, and they happen every 10 to 30 minutes. So that's the beginning of it. That's when it's like, hey, it's time to go to the hospital. Now, accompanied with dilation, usually they say, oh, my water broke. What they're talking about is the amniotic sac itself inside the uterus has broken. The sac itself has a hole in it, and amniotic fluid comes out. That's a sign that the baby's coming. Now, if the amniotic sac gets a hole in it prior to dilation, the danger of that is if now there's a hole in the amniotic sac where the baby is, but there's no labor, that puts the baby at risk of infection, because now that protective area is open, but the baby is just hanging in there still. They sometimes will actually force labor to happen. They'll administer certain drugs to make labor pains happen and to get the baby out as soon as possible. But typically associated with dilation is that water breaking. Next up is expulsion. That's where there are significant contractions of the uterine muscles. We're talking those labor pains lasting longer periods of time and happening every few minutes. That means that the baby is going to be expelled out. And this can sometimes just be, you know, 1 or 2 hours. It can last a lot longer in some women. But then the end result of expulsion is delivery of the baby. This is when the baby has actually exited the birth canal and is actually born. So here are some delivery problems or issues that can come up. An episiotomy is if the baby coming through the birth canal is too much for the birth canal opening, for the vaginal opening to bear. Instead of risking a tearing of the perineum, meaning the tissue next to the vaginal opening, instead of, you know, having that tearing, the doctor will make incisions in that region to just make it easier for the baby to come through. And right after delivery, they'll sew up that area, which is a lot easier to repair than just, you know, torn tissue. A breech birth means that the baby, instead of it being head down, the legs or some other part of the body is closest to the cervix. You can't deliver a baby normally with feet down. It doesn't slide out the right way. And what they will try to do is try to manipulate the baby from outside of the mother's abdominal region. They will try to get the baby to flip over so that it isn't a breech birth any longer. But if they cannot get the baby to flip over, they'll do a C-section or cesarean section. The other things that can happen that I didn't list here is sometimes the baby will get tangled in his or her umbilical cord. And the problem with that is if the baby gets tangled in the umbilical cord and is pressing on it with their body, they're cutting off gas and nutrients to their own body, ironically. And that means you got to do an emergency delivery, typically a C-section, to get the baby out before it suffocates. But a C-section is where they simply, they administer anesthesia, of course, but they cut into the abdominal area of the female so that they can see the outer wall of the uterus, and they cut into that, and then they take the baby out. And you can see right here, that's what they're doing. It looks kind of uncomfortable for the baby, like they're just pulling it out its head, but you can see that this nurse is assisting the doctor in getting that baby out. Premature delivery is if, prior to that, the end of the third trimester, if the baby comes out really early, like labor pains, in week, let's just say, 35 or week 30 or even late 20s. The problem with that is if a baby is delivered at the end of the second trimester or very early on in the third trimester, the risk of the baby dying is very high, because when they come out, 2.5, 3 months early, a lot of their organs are underdeveloped, and their size, their extremely small size relative to a week 40 term means they're going to be losing a lot of heat, and the baby is very susceptible to infection and such. So premature delivery doesn't mean there's definitely a problem, but the earlier they come out, the worse it is. And I mentioned with the respiratory lesson that one of the many problems with an early delivery is the lungs are underdeveloped and the baby hasn't produced enough surfactant yet. And that's that chemical that helps keep the alveoli expanded so they're not collapsing. So a lot of times they will administer surfactant to that baby to help the lungs function normally. Conjoined twins means that early on, what can happen with that blastula, with that initial ball of cells with a hollow region, is sometimes it just splits apart. That one, fertilized egg, which has become a ball of cells, splits. That's identical twins. They call it monozygotic twins because they came from one zygote, from one sperm, one egg. But if that separation, that cleavage doesn't completely finish, that's conjoined twins. It used to be called Siamese twins, but the better term is conjoined twins. They can be conjoined just, with parts of the skin, which would be very easy to surgically separate them. But oftentimes they're sharing organs. They might share a liver. I actually saw in the news recently that there was a set of conjoined twins that basically look like they have one body with two heads. They, of course, have two different spinal cords that fuse at the bottom. And I believe they said that they had two different hearts. That kind of surgery, since they are so connected intimately with their organs, they're not going to be able to surgically separate them successfully. So some conjoined twins, depending on what organs they're sharing and how conjoined they are, they can separate them. Other times it's not possible without death happening. Besides conjoined twins, I just wanted to mention the other way that twins occur. Dizygotic would be two different eggs have been ovulated, two unique eggs in terms of their genetics, two different sperm fertilize them. The majority of twins are definitely fraternal twins, not identical. And the chances of fraternal twins being identical is like 1 in millions. Just like how any two siblings born separately, maybe years apart, they'll have some things in common, but they came from different sperm and different eggs. The chances of having identical twins is something like 1 in 89, approximately. The chance of having triplets is 1 in 89 squared, which is interesting to think about. That's 1 in 7,900 and something. And then quadruplets, 1 in 89 cubed. That's a lot less likely, of course. Embryological conditions and disorders. So here's the first one I listed is one you probably have never heard of. Gestational trophoblastic neoplasia. What? So GTN, what is this? Well, the trophoblast that surrounds that initial blastula of cells, the way the trophoblast functions in terms of it growing rapidly, invading tissues of the mother, the endometrium, and not being detected by immune cells, it kind of mimics cancer cells if you think about it, what it's actually doing. So sometimes, it's rare, but sometimes that trophoblastic region will act like cancer and start invading maternal tissues. In that case, if they detect that early on in the mother's pregnancy, they have to remove that tissue as fast as possible, and they will administer chemotherapy. So of course, the baby's not going to survive that, but really weird thing that can happen with that trophoblast region. A miscarriage or spontaneous abortion means through no active choice of the mother. It just happened randomly. A miscarriage can occur early on in pregnancy, even a little later in pregnancy. So spontaneous abortion, the mother didn't want it to happen. It just happened. And there's lots of reasons this can occur. Lots of reasons the baby didn't successfully develop to full term in the mother. Of course, drug use or alcohol abuse can increase the chances of a miscarriage. Induced abortion is the technical term for when a mother elects to actually get the baby taken out of her and terminated. Ectopic pregnancy is not very common, but if a woman of childbearing age is sexually active and there's even a chance that she could get pregnant, the first sign of an ectopic pregnancy is a really severe, sharp pain down in the lower regions of the abdomen. Not exactly where you would have the pain of an appendicitis, but not far from there. And that's a sign of an ectopic pregnancy. What this is you'll have fertilization, and that ball of cells, that morula or blastula, it's supposed to roll down from the fallopian tube into the uterus and implant itself there. Well, if that ball of cells implants itself in the fallopian tube and starts growing, eventually that's going to result in a lot of pain, because the actual diameter inside of the fallopian tubes is only a few millimeters. So early on in pregnancy, the stretching that fallopian tube is extremely painful, and the baby is not going to make it. But if that's not dealt with, the bleeding resulting from that could kill the mother. So this requires an emergency surgery to remove that particular embryo. In vitro fertilization is what a couple will do if they're having trouble with the fertilization in vivo. In vivo means, in a living being. So natural sexual intercourse, natural conception is in vivo, but in vitro, if a couple's having trouble, they will take sperm from the man, eggs from the female, mix those together in a laboratory, like in a petri dish. Then they'll get a few little embryos and implant them inside the mother's uterus. They will implant a few, hoping that just one will take, that one will successfully be implanted in the endometrial lining and develop a placenta, etc. But oftentimes, multiple ones actually implant and make it to full term. That's why the chances of having twins or triplets or quadruplets, etc., is much higher with in vitro fertilization. Amniocentesis is one of the tests you can do to see if there's birth defects early on in the pregnancy, if there's chromosomal problems. And they recommend this for women who are in their 40s or later on, or if there's a history of birth defects in your family. So here's a normal karyotype. Here's what they do. They take a needle, go into the amniotic fluid through the uterus, and withdraw some of that amniotic fluid that's surrounding the baby. In that fluid, you're going to get skin cells from the baby that have sloughed off. So many of those. So in the fluid you withdraw, there's bound to be some of those skin cells. You extract the DNA. You can actually before extracting the DNA, you can stimulate those cells to undergo mitosis, so you have a lot more DNA to work with. And when you take out the chromosomes from a cell and they're laid out in pairs, the pairs that go together, there should be 23 pairs. And you can see there's all the numbers 1 through 22, and here's the 23rd pair. So this would be a male karyotype because here's an X chromosome, here's a Y chromosome. If instead of XY it was XX, well, then we know it's a female. So looking at the chromosomes, they can diagnose certain chromosomal disorders from this particular amniocentesis test. Speaking of this test, you can detect certain birth defects. Down syndrome would be, if you look at pair 21, Down syndrome is caused by trisomy 21, meaning 3 copies in this particular part here. You're supposed to only have 2 copies of the 21st pair. It's a pair, but having an extra one either from the sperm or the egg, and that happens through non-disjunction errors during meiosis that you may have learned about in biology. Down syndrome, has forms of mental disability. There's a certain look on their face if they have Down syndrome, probably because chromosomes associated with this pair have something to do with formation of the skull bones. But yes, Down syndrome doesn't have a cure, but individuals with Down syndrome can live normal lives. Turner syndrome, rather than it being on the 21st pair, that's actually affecting sex chromosomes. So all of these 1 through 22, I'll make a box around them. 1 through 22, you've got autosomes, meaning they're non-sex chromosomes. So if you're a male, there's a female out there on planet Earth who could have an identical, chromosome 20 to you, coincidentally. But she looks like a female and you look like a man. So only these right here, chromosome 23, directly pertain to sex, being male or female. Turner syndrome is when you're born with an X chromosome and no Y. So they are female, but underdeveloped in terms of the ability of them to actually have kids, and their genitalia is underdeveloped. And they usually don't look like a typical woman when they go through puberty. Spina bifida basically means split spine in Latin. Spina bifida means there was a problem with the development of the spinal cord and it being completely encased correctly in the spinal column within the back. So spina bifida can be very minor, meaning the spine is enclosed inside the back correctly and protected, and there's meninges around it, but sometimes parts of the spinal column, meaning the bones, don't completely encase or ensheath the spinal cord. So those people are a little bit more at risk for certain injuries to their spinal cord because it's not completely protected. The worst end of it is a baby born with severe spina bifida could mean that part of the spinal tissue, the spinal cord, is actually outside the body. And that's huge risk of infection, and the baby typically doesn't live long with that. PKU is one of those few birth defects that if treated correctly, you will see no negative effects. PKU stands for phenylketonuria, which means you were born with a gene, a dysfunctional gene in terms of your ability to break down phenylalanine. So phenylalanine is an amino acid in the body, and if you have PKU, phenylalanine in the body can build up and cause brain issues, brain problems. But if you know your baby has this, make sure you don't feed the baby phenylalanine, and then you will not see any long-term effects of PKU, which is pretty amazing. And you can look up a list of what kinds of foods have a significant amount of phenylalanine. But anyways, these are only 4 birth defects in thousands of disorders that can happen. So if you're healthy and you don't have birth defects, be thankful. So thank you for watching Educator.com.